I am a freelance journalist that has traveled to Kampala, Uganda to learn about gene editing at the National Agriculture Crop Research Institute. Doctor, can you tell me about the work you're doing here?
Of course. The emerging fields of synthetic biology, nanotechnology, and gene editing have been making waves in the global scientific community recently, and this includes scientists working in different laboratories in Africa. Scientists are already applying the technology of gene editing, nanotechnology, and synthetic biology across all fields of agriculture. In many cases, there's not even an introduction of foreign genes, which is also the focus of criticism for anti-biotech groups who claim that gene modification alters natural biological states. These technologies do not require a new gene from the plant for resistance. For example, nanotechnology for breeding of cassava, a tiny gene is obtained to mimic the cassava mosaic virus. 1 millimeter diameter of the mimic gene can yield about 2,000 more millimeters, which can help identify the organisms causing the disease. At the end, we get the recombinant genes in the breeding process to resist the genes. There have been controversies surrounding the application of genetically modified crops, but now more than 90% of certain crops in the U.S. are genetically modified.
So what is gene editing?
For example, here we're dealing with a plant who has a gene that's retarding its growth. We want the plant to look like this, nice and tall. These are our two strands of DNA. They're almost identical, except for these two genes. These two genes are the genes that are retarding its growth. So in order to make the plant grow like this one, we're going to silence these genes. Now, these two strands of DNA are identical, and the plant can continue to grow.
Every single behavior of a plant is controlled by a gene. When a scientist identifies a stunted plant, he or she can silence this gene in the laboratory to enable the plant to grow tall. Or you simply delete the gene and eliminate it in the life cycle of the plant. There are genes which control disease resistance in the plant. It is the duty of scientists to ensure these genes are expressed for the plant to resist certain diseases for it to grow normally. We are now moving away from sourcing genes in other organisms to be inserted in plants that are challenged for resistance. This is a close-up of what we showed previously with the doctor in the lab.
Scientists breeding mosquitoes for malaria prevention are already using synthetic biology in the breeding process. This is because of the 300 species of mosquito in the world, those causing malaria are probably only 2 or 3 species. Scientists have silenced the gene that causes reproduction during the mating process. This led to the disappearance of females in the environment because the females are mainly the ones responsible for spreading and causing malaria infection in human populations. Scientists in Uganda at the Virus Research Institute with their partners in Senegal are already carrying out research in the same field.
After speaking with scientists, I've learned more about genetic editing and synthetic biology. I find this technology imperative for advances in agriculture. Seeing how genes are now being silenced, this is not only going to help agriculture, but also all aspects of